Great job, Laura and Maria. Try to share my screen now. Do you see this orchestrating microbiome analysis?
Yes, I can see your screen.
Okay, that's great. Then it is the correct one. So hello, my name is Tuomas, as it was said. I had this introduction slide also, but most of this was already covered. So I'm a doctoral researcher at the University of Turku in Turku Data Science Group. So if you know Leo Lahti, he is my great instructor, pretty nice guy. I have been living all my life in Turku, and that might indicate that this is the best place in the world. I don't know about that, but it's pretty nice place. Then, well, I'm currently developing microbiome tools in Bioconductor, and maybe shortly about my education. So I have a degree from biotechnology, so I know a little bit about what people do in the wet labs, but not much. So I will give those questions to other people if you have those. And then I have also degree from business, but that was, if you know those video games, there's always this side quest. So that was, I guess, side quest in my life. I'm far from a business guy, more like engineering guy. Maybe in the future. Let's see. Yes, so that's what I'm currently doing. And as I love to market things, the European Bioconductor Conference, as it was said, it is in Turku this year. In 25 days, it will be happening. The late breaking posters submission is still open. So if you want still to come, you are very free to join. Of course, it's pretty expensive to fly here to Northern Europe, but we are at least recording those talks. I recommend to check them later from YouTube. Great. So that was the introduction to my life, but that's not the topic of this webinars. Instead, we are talking about the microbiome stuff. In this session, I planned to talk about ecosystems, microbiome data science ecosystems. So there are several of them. Bioconductor, and especially this that we have developed is only one of them. So we will cover a little bit the background of these. Then next week, I plan to go through the end-to-end workflow. So how we get the data, what we do to data to get the abundance tables, and then what we do in the statistical analysis part. And this week, the last session is mostly covering the statistical analysis part, that part that we are especially developing. Great. So about the ecosystems. Bioconductor is a community-driven project, global project, and it is very well known for the software. But I would say that it's the main point of Bioconductor is not the software; it's the community of people, developers and the users. Of course, this is kind of the question of that, if you know the chicken and egg, which came first, the community or the software. But I would say that nowadays, when we have this very nice community of people, it's much easier to think about the future development of software. This Bioconductor started 25 years ago. So this is anniversary year, and it has very high impact in bioinformatics. There is 50,000 citations in PubMed and 1.5 million downloads of packages from distinct IPs last year. 1.5 million, that's a very, very big number. Then I will let you to read this. I will give you 20 seconds to read this text. I will take a little bit water now. So you might wonder why the heck I'm talking about alchemy and alchemists. But I have a very good point here. So what those alchemists did in Middle Ages and after and before that was that they tried to do expensive materials, gold, from inexpensive materials like iron and urine. And of course, if you think about it, you cannot really make gold from urine. And they did lots of studies, lots of experiments, and they tried hard. They tried very hard to accomplish their goals. And of course, because it's not possible, they failed a lot. But instead of thinking about if the theory of making gold from iron is correct, they were criticizing instead the laboratory equipment. They were thinking like when they were failing like that, oh heck, again the fire was not warm enough or something. I don't know, but they were complaining about the tools and not the theory. They were not thinking about if the theory was correct. And lots of these experiments, when they, of course, they were motivated to do this stuff because many of them, those studies seem to work. But of course, they did not replicate those results anywhere. And why this was rolling this loop was that they didn't have this kind of community, critical community, that really thought that, okay, we should probably think about again the theory. And in, I guess, 1700s or something, then the chemistry as a field of science was formed because they started to really think about the theories, and they had this critical community developing these ideas and theories in science. And the same goes with bioinformatics. So that is why we have this open communities, such as Bioconductor, where we discuss these things. What is the methodological theory, and what is the try to form the best practices and so forth. And that's why we have the open open source code. That's why those journals, fortunately, they nowadays require that you give the source code of your analysis and data if possible, because the open communities, open critical communities are the key to success, not those closed communities like Alchemist did. And these open communities are the ones that drives towards the quality and high impact. Hopefully, you are now motivated to do open science. And how Bioconductor does this open science is with this open source code. So all this software is openly available. So you can always go to check what they are actually doing. And if you think about that something is wrong, you can create issues to GitHub and discuss these things. And everyone can contribute on that. Then related to the replicability, there is this documentation that shows how the methods should work and how people should use them. And also this community support is very important. And these kind of events that we are now in this webinar, they are very important in forming this community of people to progress this bioinformatics field. This kind of open microbiome data science ecosystems, there are more than Bioconductor. There is several of them. For instance, Motur, then Chime 2, and Anvio. So Bioconductor is just one of them. And they differ from their scope and also the community, but they are also very much overlapping. So for instance, Chime, while the Bioconductor is in statistical analysis, in Chime you can do the taxonomy mapping and then the statistical analysis. But all these different ecosystems, they have their own pros and cons. And in Bioconductor, one of the big things is that the aim to make the software more interoperable. So across these ecosystems, you can import data from Chime and then use the Bioconductor tools, or then from R to Python, for instance. But of course, as this is a Bioconductor webinar, we are talking about Bioconductor, but these other ecosystems, they are, of course, I recommend very much to use Bioconductor, but you should be aware that these other ecosystems also exist. So little summary of Bioconductor software or those packages. So from this left figure, you can see this number of packages through the years. So you can see that the number of packages has grown, and currently there is 2,300 around that number of packages in Bioconductor. And from here you can see also how they are distributed to different fields. So genomics is the one of those biggest ones, and then there is microbiome is the red one. So it's a relatively small, but that's also one of the key advantages of Bioconductor compared to the Chime and other ecosystems. So this Bioconductor is much more than microbiome, and that's why there is experts from different fields of bioinformatics in Bioconductor community. And one thing about Bioconductor is that the strict requirements of Bioconductor package. So every package goes through the review, and then, when they are submitted to Bioconductor, and if they pass those that review, they go to the Bioconductor, and every second day, if I remember correctly, they undergo automatic testing so that they are working as expected. And third point about Bioconductor packages is that the requirements about the documentation. So there should be reproducible examples, and they should be thoroughly documented what the methods are doing and what they should do. And as I was mentioning last year, this is the right side figure is showing the number of downloads from distinct IPs per year. So it has grown a lot, and I don't know what happened last year, but now it's almost 2 million downloads. So it is growing fast. And in Bioconductor alone, there is this several ecosystems. So this phylogic, how many of you know phylogic? You can, I don't know if I can see, but there is this button that you can raise your hand. I cannot see anyone. Yes, now I see. Great. So phylogic is one of those microbiome ecosystems in Bioconductor. And it was developed around 15 years ago. It's very good ecosystem, very broad, very mature. I really like it. It was developed for because in that time the 16S data, so the amplicon sequencing data, was the main source of microbiome data. We will go through this later, what they mean, this amplicon sequencing, but phylogic was developed at that time. And because of that reason, there is certain limitations in this phylogic, which this Treesummarized experiment is aiming to handle. Then there are this summarized experiment that was developed after phylogic was established. So this summarized experiment is generic data container in Bioconductor. So it is tailored for generic purpose container for genomics to proteomics to metabolomics and so forth, for every kind of this kind of data that you might have in biology, in computational biology. And then it is extended, this same data container, to single cell and then to microbiome field with this tree-summarized experiment. Now you might wonder what is data container, but we are going through them very soon. So here the main message is that there is several ecosystems in Bioconductor, but then there is this main data container, this summarized experiment, which has been then extended to different fields, to single cell and to this microbiome field with this tree-summarized experiment. And because they are extending this summarized experiment data container, this all ecosystem, single cell and microbiome field, these all are interoperable between each other. So that is the main point. Phyloseq is kind of alone because it's just a Phyloseq, but when we are using the same general purpose data container, we can interoperate between different fields. So this tree-summarized experiment was not developed by us, it was developed by Huang and others. And we then used this tree-summarized experiment to create this our ecosystem of methods. So this data container alone is like a car without the engine. So we created the engine to that car. So the engine is this orchestrating microbiome analysis ecosystem, and it is for downstream part of this microbiome workflow. And by downstream, I mean the statistical analysis part. So upstream is then the taxonomy mapping. So when you have the sequences, so what to do with the sequences, you map them to some taxonomy to get the abundance table. So that's upstream. And then when you have the abundance table, then you do statistical tests, and that's the downstream. So this ecosystem is tailored for this downstream part. And this microbiome analysis MIA package is the core package. It includes these transformations, alpha diversity, beta diversity, and other metrics. And it has been last year it was downloaded 15,000 times, and it's top 8% in Bioconductor, so quite popular package. And then there is a preprint available on this ecosystem. So if you think about this, now the Bioconductor ecosystems, we have this that we have the orchestrating microbiome analysis, this ecosystem, then we have Phyloseq, then we have others also in Bioconductor. So what is the benefit of using this our ecosystem is that I was explaining how the summarized experiment is extended and how then tree-summarized experiment, because it's extended from summarized experiment, how that is then interoperable. So that is the first point. We can use the same methods across different fields, and that makes it tightly linked with other Bioconductor ecosystems. So for instance, we can borrow methods from single cell field. Then one thing is that it improved scalability. So those data sizes has increased in microbiome field, So how to manage this kind of data that has thousands of samples and thousands of features? You need system that is capable to handle that kind of big data. So that is also one benefit of this ecosystem. And because this data set, this ecosystem is designed for multi-table data, it's inherently it can handle this multi-table integration. So if you think about microbiome data, usually we have these raw counts, then we have transformations on that same table, but then we can have also metabolomics data, for instance, or shotgun data, we get the functional annotations. So when we are using this data container, this Treesummarized, this ecosystem, this can be done in an efficient manner. Here is the list of different packages in this ecosystem. So Mia is the main package. It includes the core analysis tasks. And then there is miaViz for visualization, miaSim for simulation, miaTime for time series analysis, and then others. But the point is also that this is not only our work, not our team's work. It is a community effort that has been done. So there's lots of independent developers that support this Treesummarized data container, which means that there is lots of tools available for user to analyze the microbiome data. And one important thing is also that there is this online book available. So the online book, the aim of that is to promote the best practices in the field. And of course, those best practices are not formed by us, it's formed by community efforts and the publications. So the evidence, scientific evidence. And that's why we have lots of citations there in the online book. It shows how to conduct the analysis. So for instance, calculating alpha diversity metrics and then how to visualize them. And because this is a community effort, it is open to contributions as well as the packages. We are very happy if someone is willing to give feedback or create the issue if something is not working, or then develop something new. Also, discussing in the forums is very important. So that, for instance, just discussing what you are working on and how we can help in that. So that is also very important. Now I will show the book how to find that. So it is available. Here is the Bioconductor homepage. So from that homepage, you will go to the Bioconductor books. And then there is several of them, and then there is also others. If this is Orchestrating Microbiome Analysis with Bioconductor, the microbiome book, you will see that there is this Orchestrating other omics also. But let's go to the microbiome book. So it will look like this. So from here, you can find lots of information on this ecosystem. and how to conduct the data analysis. So for instance, if we go to the diversity and then alpha diversity, you will see that there's lots of information. We cite to the newest publications, which, for instance, this publication is suggesting to calculate these matrices. So that's recommendation, best practice, and then you can find here examples on how to calculate that those matrices and then visualize them. So the idea is to show how to do that in practice, but also to disseminate the best practices. So that was the in brief. I will still have material to show today, but this was the introduction to the ecosystems. So in microbiome field, we have several ecosystems. We have this chyme, mothur, anvil, then bioconductor. And inside bioconductor, we have also several ecosystems. For instance, phylocyc and this Treesummarized based ecosystems. And in these sessions, we are going to focus on the Treesummarized based ecosystem. I was mentioning the data containers. So that will be the next topic today to cover. That's very important to know what they are and how they work. What is the idea of data containers? Because they are, I agree that they can be a little bit complex at first, but when you know how they work, it becomes very handy. So data containers. And data containers, in Bioconductor, the core of software is formed by these data containers. And what they are structured way to present and manage complex biological data. So for instance, if you think about in biology, like a common analysis, we have the abundance table, one table, and then we have the taxonomy table, then we have sample metadata, then we can have metabolomics and so forth. So managing this kind of complex data set with many tables becomes quite hard, and they become the bottleneck of efficient workflows. So in this figure, data container is in the middle. They are the core, and when those developers agree this standardized data presentation, data container, they can develop methods that are utilizing this very specific data container. And when they are developing methods, lots of methods, that ultimately means that the users and the community can then, when they put the data on this very specific way, data container, those community have more and more tools to utilize, to analyze their dataset. So data container is the way to enable this efficient and modular workflows. So with user perspective, we put the data to this data container, PhyLogic or Treesummarized experiment, and then we can apply methods that are utilizing this data container. So this is a very simple example of data containers. So here on the left side we have our office with lots of different papers lying around. They are unordered. You don't know where to find what page. And then on the right side, we have this very harmonized way to present the pages, so-called book. So you know that in this book, you have the table of contents where you can search where to find the specific information, specific page. And you know that after page 1 comes page 2, page 3, and so forth. So that is very harmonized and structured way to present many pages of paper. So now you could think about whether you would read 100 pages from this left side, this mess. You don't know where is which page, and you have to find them. Or would you read 100 pages from this data container called book? And hopefully everyone is agreeing that you would take this book. And the same goes with these data containers. So in microbiome field, you can have abundance table, sample metadata, taxonomy table, phylogeny, and so forth. So instead of analyzing them alone in a separate data types, you will put them to this data container. And this data container is very structured way. You know where you can find, for instance, sample metadata, and that does the linking between samples. So if you select one sample from this dataset, it selects them from abundance table and the sample metadata at one go, because they are linking these different data types. In this separate in the left side, this does not do that. You have to do that manually, and then you end up doing mistakes. So that is the idea behind data containers, why Bioconductor utilizes them, and why they are in the core of this software. Because that makes easy to manage these complex datasets. And when we agree the data format, then we have many methods that we can apply to analyze the dataset. But what is the optimal data container for microbiome data? It must support multiple assays. It must have the linkage between the different data types. So as I was mentioning, the sample metadata and the abundance table. But then we can have also the raw counts, then transformations of that abundance table, metabolomics data, proteomics data, and so forth. So the optimal data container must support these different tables. Then it must support hierarchical data. So bacteria and microbiome, they are usually presented as a phylogeny. So we should have a support for these phylogenies, but also samples can have some hierarchy. For instance, if the samples are collected from people that are relatives or something. Then it should have support for site information, so taxonomy table, site information on the bacteria, so from which class or family they are from, and also the sample metadata is a site information. Then the size of the data has increased in past years. It's not the seventh wonders that if you found some data set that has like tens of thousands samples, that normal day nowadays. So these data containers must be efficient in speed and memory. And one big thing is that if you are familiar with Phylosec, as I was mentioning, it was developed in the era of 16S data. But nowadays, we usually have the shotgun data. We are going through that next week, what these mean, what is amplicon and what is shotgun data. But from shotgun data, you will get, in addition to the taxonomy mapping, so which bacteria is present and how many times, you will get also the functional annotations. So you will get these many tables, and then quite commonly in studies you have metabolomics data and proteomics data and so forth. So this, when we are using this Treesummarized experiment that I was mentioning earlier, we can use the same methods from metabolomics field because they are using the same exact data container. So we can use the same methods to analyze the same data container to analyze the taxonomy data and also metabolomics data and proteomics data, and that's one of those big advantages of using the Treesummarized experiment. And also this means that because we have now harmonized way to across fields from metabolomics and this microbiome field. We have the same data container. We can collaborate with them, and we have much more knowledge than to analyze these complex multi-omics datasets. So these are the requirements of optimal data container. And surprise, surprise, it's called Treesummarized experiment. That is the optimal data container for microbiome data. Let's go through this the structure of this Treesummarized experiment. So the core of this data structure is the abundance table. So how many times each bacteria is found from each sample. And then the cells of this table is some numeric value. So that is found from here, from essays. And you can see that there is 3 different essays. So for instance, this can be the raw counts, then this can be logarithmic transformation. This is, for instance, CLR transform data. So this tree-summarized experiment can handle multiple abundance tables. Then in columns, we have samples. And then in the call data, column metadata, we have the sample metadata. And this, you can see this arrow and this coloring. This means that these are linked. So if you select, for instance, this sample, this automatically selects the sample from the abundance table and the sample metadata at one go. Then in the rows, we have bacteria. And the same goes with bacteria. We have the row metadata, which is often the taxonomy table. So these abundance tables are linked with this row data. And then we can have for both columns and the rows, we can have hierarchy tree. And this column tree is not that often used. I was mentioning the relativeness of patients, but that's not too commonly used. But this row tree is very commonly used because this is the phylogeny. And that's usually the information that we want to analyze. So that can be found from a row tree. And this row link and column link, they are just linking these nodes from tree to these rows in this, or this bacteria in this dataset. So for instance, these two bacteria here, because they are orange, they are linked to the same node in a phylogeny found here. And this bacteria here is linked with this node right here. And this metadata, that can be just generic information who collected the dataset and so forth. So this is the tree-summarized experiment. It is important to understand the structure of it. And when you understand the structure, it's as I said, it might seem very complex first, but when you understand the structure, then you can use it to analyze your microbiome data, but also the metabolomics data if you have that, or proteomics data. Because as I mentioned, this tree-summarized experiment is extended from summarized experiment. And this same data container is used in a wide variety of fields from genomics to metabolomics and transcriptomics and microbiome. So this is more or less what I wanted to cover today. So one point was the first section of today was this community. So community is the very key. Otherwise, if we are doing our analysis closed source, we don't give the source code what we have done in our analysis, then we are kind of alchemists. So that's why we are doing things in this open community, and we are discussing things openly, and that is the key of success. And then there is several different communities and ecosystems And in Bioconductor, we have this Treesummarized experiment-based ecosystem. And this Treesummarized experiment was the data container, the structured way to present and handle this complex microbiome data. Great. These was the words that I wanted to say today. Do you want Laura to take the mic from me?
Yeah, sure. Thank you so much, Tuomas, for introducing the microbiome analysis using MIA during the session today. Just in case anyone in the call doesn't know, this is going to be a series of sessions. Today we have the first session, and then next Wednesday we'll have the second session, and the 20th of May we'll have our third session. So today Tuomas was introducing, and I like the points where he said community is key, obviously, and introducing the Treesummarized experiment. So we will be taking questions now. Feel free to unmute and ask directly, or put your question online, beginning line 72 of the Frama card, which I have been posting on the chat. Yeah, and I don't know, whoever is typing the question, do you want to unmute and just ask directly, or should I read your question to Tuomas now? Okay, no one is unmuting, so I'll just read it to you, Tuomas. So the question asks, are there any libraries that extend the summarized experiment that could concentrate the data into an embedding and visualize the data with t-SNE or any other visualizations?
There is, and there I cannot remember, was there, for instance, that can be found from single cell package called scatter. And we had examples on that t-SNE method. We had examples in the online book, but this more simpler principal component analysis, we can cover that in the next sessions, but these are more recommended nowadays. Principal component, principal coordinate analysis, and this information can be found from the online book, and we can also cover them in the next sessions.
Well, thank you. Does anyone else have any other questions? Also, just a reminder, if you've not put your name on the icebreaker session section on the Frama card, please go ahead and do so. It will really help us with our note keeping and record keeping efforts. Yeah, thank you. Okay. If there are no further questions, we'd
Hello.
Like
Hello.
Yes, Pascal, go ahead.
Okay. So greetings, everyone. So I thought that this session was going to include maybe using an R package to maybe do some analysis of metagenomic data for us to have a feel of how this the bioconductor package was like. And secondly, I have some interest in is it genomics? Genomics, and I'll be very glad if the facilitator for today can maybe recommend our packages that can be used, specific packages that can be used for genomics. Okay, thank you.
Yes, good questions and points. So we have 3 sessions, one this week, and then one next week, and the third one 2 weeks from now. And this session was aimed to form the basis of this. And if you don't know what is the ecosystem, then it's very hard to continue from that. And the next week idea is to start to do the and go through what is we actually doing in practice. So what is where we get the data, what to do with the sequence level data to get the abundance tables, and then how to import the data to this Treesummarized experiment that I was explaining, and then how to analyze that. And the third session is only for showing how to analyze. So there will be information on how to conduct the analysis in practice. And for genomics, most of these methods are pretty generic. So most of these can be used also in genomics, but I haven't done that too much. I mostly do metagenomics, so I cannot give you exact answers to that. But most of these methods are also can be used in genomics.
Thank you for clarifying that, Tuomas. So one last question, then we close this. It was only supposed to be an hour, and we will have more of the coding demonstrations in the subsequent sessions. So Sarah has left a question on the chat, and I'll just read it for the recording. Is data containers can be compared to micro table objects from micro eco package?
Yes, I know very well the micro eco package, and I cannot remember exactly the details in that manner that if this is similar to micro table, but I assume that the idea is very similar. But as I was mentioning, the phyloseq, it is only for microbiome data, and the same goes with this micro eco package. So the limitation, they have very nice ecosystem in micro eco people. They have built very nice ecosystem there, but the limitation is that they are not using this summarized experiment that we are using, that is used across Bioconductor in more generic from genomics to metabolomics and so forth. But yeah, I assume that the idea of microtable is very similar to these other data containers.
Well, thank you. I hope that answers your question, Sarah. So we will stop here. Thank you everyone for joining the call. If you're already registered for this session, we will just be using the same link for subsequent sessions, but we will be sending you a reminder just in case you forget. Also, if you still want to join our Zulip and sign up for our mailing list, those links are on line 87 and 89 of the FromUp pad. Please come join us on that other side as well. We usually have some, updates there. So at this juncture, I will just hand it over to Maria, who's our community manager for Bioconductor, probably to say a few words and how this session is possible. Welcome, Maria.
Thank you, Laura, for facilitating and leading this session, and thank you everyone for joining. We're just a couple of minutes over, so I just want to say thank you again to Tuomas. That was a really clear and helpful introduction and a great foundation for the next sessions, and I'm looking forward to parts 2 and 3. So yeah, we will see you all next week. Thanks.
Thank you, everyone. See you next week.
Thank you. See ya.
Thank you.